Hello, my beautiful friends. So, this is our continuation with the lesson 2 after a short introduction. And in this lesson, we will talk about latest science of aging explained. Okay, so let's get started. So, this is our second part of our 8-part mini course, Awakening to Vitality. And we are about to talk about latest science of aging, how it actually, all this stuff works, what are the truths, what are the facts, what are the myths behind it, what actually works. So how actually the mechanism of biological aging works, what those latest research shows, what are the things behind information theory of aging, which is a completely new approach to aging as a process from scientific basics, and how actually the biological age is measured. So we will start with a brief review of our understanding of DNA. It's a basic database of all your genes in your body. Every cell of your body actually has the complete DNA in it as a complete plan how to build the whole body. But it's important that in every cell you have a different expression of your DNA. So your DNA has just a small bit of the data available to build up your individual organs, your tissues. So we are returning back to a little of history, how actually the aging has been understood just several decades before. First of all, it was in 1950s, 1960s, called as a mutation theory of aging, where Peter Medawar and Leo Szilard, those great scientists, even Nobel Prize rewarded, understood that aging has to do something with the DNA quality of somehow mutations or any damages on DNA has been accumulated, and therefore your body couldn't rejuvenate, and your cells are not replicated properly. After all these decades came a new theory where it started to talk about antioxidants. So some possibilities that you can even reverse this DNA decay or DNA damage with a special substances, special molecules, which one of the most important was found resveratrol, which also was basic for so-called French paradox, that actually even French people which do not have a perfect nutrition, eating a lot of fatty food, but drinking a little bit of wine every day, are still having almost perfect health in comparison to other citizens which in other countries which do not drink any red wine. So resveratrol was started as the most important antioxidant. It boosted also selling of the red wine maybe by 300% from the time from 2002. And the understanding was that actually the stressed plants, in this time it was your wine, was starting to produce special xenohermetic substances which tells your body that it needs to be prepared for starving, for starvation, for bad times. What was a follow-up with this method, with this theory, was that understanding mutation was disqualified as the basic reason for aging of our body, because in 2013, great scientist Leo Garente from MIT started to understand aging with a simple yeast. Yeast as a small bacteria from which every bread is made. And he found there are unbelievable correction mechanisms in every living cell on this planet. So actually, he understood that every cell from plants to yeast, also in the human body, has so big abilities to correct your DNA that actually it's comparable to autocorrection in several thousand times on every cell every day. So you can compare it and understand your DNA as a basic database of your founding plans, of your building plans, as some installation CD of human hardware and software. Every human body has around 4 billions of nucleotide pairs, GTAC, and every cell, individual cell, has a golden master CD, which is pretty same throughout the whole of your life. Only different on every cell is that it's a separate expression of this installation, and we can understand it as the separate installation of the operating system or separate implementation. This is how the biological cell looks like. So you can see on the left side the nucleus where the DNA is actually based, and every cell has to replicate several times, many times. Sometimes it's several tens of times throughout your whole life. And to be able to build correct peptides or building material for a new cell, they have to somehow take all this correct data from DNA with the mediatory RNA, ribosomes, and from ribosomes, actually, you are creating a specific chains of amino acids, which then later looks as a proteins covering your skin, proteins creating your tissues, your eyes, your hair, or every organ of your body, or even every hormone in your body. So actually, what's going wrong with this replication process, with the aging? We understood in the last decades that actually the implementation is something like analog data, which represents installation of DNA, which is completely untouched throughout the whole life. Otherwise, you would be not able to have healthy children in your 60s, or many people are having children in very late age. So it means the DNA has been replicated almost without any impact, any damage. And Leo Szilard, we already talked about, and Leo Garente, that every cell actually loses its identity. So despite your cells do have all the data, they are stopping to understand, or they're losing their memory of what was the original differentiation, what was the original build out of these cells. And this is actually the target of the strategy we will talk in this program, how to actually revert the identity of your cells. So very briefly about understanding how actually our DNA data is separated, how it looks like. So a human genome has 46 chromosomes. You have a pair from your mother and from your father, from each. So it's a 22 pairs of X and 1 pair of Y. How you can imagine how it's actually data stored? It wouldn't be a CD in actually, it's a typical disk shape. You can understand every chromosome rather than old tape cassettes, audio cassettes, or from older time, there were also possibility to record the data, and you can have games, you can have any software recorded on every such audio cassette. So 46 of these information, which are perfectly wound, perfectly stored and packed on these individual cassettes, you can have a complete DNA of a human. What actually happened? How you can continue with this analog information? Cells need to somehow work with gaining additional understanding of this and explanation of this data from this tape. So actually, DNA needs to be somehow extracted and unbound, opened from this closed cassette, and correct bounding, correct exploration of this data makes actually the correct identity of your every cell in your body. And actually aging makes your cell losing the correct differentiation of these data which are available for reading, and the rest of the data which are closed and very densely packed. Your DNA is so perfectly supported and corrected that even after sometimes 10,000 times a day damage, your cell is able to correct every break or every damage in its structure. So these reparation mechanisms are the things which we are keeping our focus to be able to keep our cells healthy. And we will talk about those mechanisms soon. So this is actually a very brief understanding how the data are stored. And we don't need to remember much, just the understanding DNA methylation. Because DNA methylation are actually the small acetyl groups or markers on your DNA, which are packed in your chromosomes. And these methylation marks are defining what is readable from this data and what is sealed area. And this readable versus sealed area is defining how actually your cell looks like, and actually what from DNA is presented. And maybe you understand correctly, also serious illness like cancer has to do a lot with the DNA methylation, because cells are actually losing the correct markers, how the peptides and then proteins, building materials of the correct design of the cell looks like. And we are coming to a first magical switch or magical molecule or mechanism, which is almost billion years old and which has been also studied on the yeast, and it's called sirtuin, which represents also a genetic switch, which is able to silence information and actually revert and correct the backing of the data on the DNA. So it's important to remember sirtuins, and the function of the sirtuins is one of the most important strategy and supporting it to be live, to be available in every cell of your body, to sustain with a long health and longevity as well. Every living organism on Earth has it. It's maybe 1 billion years old mechanism, and these sirtuins are able to stop any other function of the cell, like reproduction, and in the bad times, they're coming back to DNA. They start to correct it every time there is something damaged or incorrect. So you can understand this methylation and the correction by sirtuins, as you can see on these audio tape cassettes, which is limiting to correct information to be visible only, and the rest to be correctly packed and stored in the nucleus of the cell. So we are coming to an important understanding of the latest science, which actually tells that that DNA methylation is the basic problem which we are coping with the ongoing aging of the cells. And this DNA methylation is also the target mechanism of the biological age, understanding, measurement, and we are trying to understand also the controlling other mechanisms which are trying to perform this epigenetic information. This epigenetic information is actually the additional data, more in analog way, in addition to DNA, which actually tells how the cell should look like, what are the correct definition of the peptides. And actually from maybe around 20 million methylation sites on our whole human genome, we are performing measurement on around 120,000 of locations or islands on our DNA. And based on these decay of these methylation marks, we are able to expect biological individual age. Okay, so the very brief wrap-up of the information theory of aging is that we don't need to update and operate with the chemical substances, with the physical problems. We are not damaged physically, rather our organism and details in every cell are losing correct information. And that's why the most of the strategies and also understanding your aging will comes from information technologies, from DNA methylation, sequencing, and also correcting of the data in your every cell is the basic thing which make you more healthy, which make you without cancer, without cardiovascular diseases and other significant problematic illness. As I said, it's a very robust scientific method which is performed on the DNA methylation machines, which at the beginning cost around $1 billion of US dollars when the whole human genome has been analyzed. And today we are able to perform analysis of the whole human genome just in several hundreds of US dollars. So we are using Infinium EPIC array, which is a separate specific array from standard sequencing. We are looking for methylation islands on DNA, and based on the machine learning methods we will talk later, we are able to predict your correlation with the older or maybe younger samples. We are using the Clock Foundation, which is the already 10 years old method of understanding of, as I said, machine learning of samples of DNA methylation with Steve Horvath, a scientist from US, which has beautiful methods for analyzing this data with the artificial intelligence, with genomic data information technologies. And they have developed also several additional measurements and indicators, for example, also pheno age and Grimm age, where you can understand your expected date of your death, even, or your total lifetime. So, as I already mentioned, Steve Horvath was the basic scientist which started this methylation age measurement. I have started my methylation measurement already in October 2022. We will have the final measurement of our methylation age in our pilot group in Slovakia by end of August. So soon I will share it also to our social group on Facebook. So we will be able to see the results if you join there. And it's first of all, one of the most precise method of understanding biological age. It's done already since 2013 on the thousands of samples from all other tissues, and you still have very good results in comparison to any other method. So this is end of our understanding of science. We will continue soon with understanding how actually the strategies of age reversals work.